Bismillahirrahmanirrahim. Assalamu alaikum. I am Professor Dr. Mahmouda Begum, Professor and Head, Department of Anatomy at the Womens Medical College, Mogbazar, Dhaka. Today I would like to start the events of first week of development, that is from fertilization to implantation. First, the fertilization. This is the figure which focus the events. These occur the region of the fallopian tube. Fertilization start in the fallopian tube, and after the fertilization, next events cleavage occur also in the fallopian tube, and the morula, when the morula form, they enter the uterine cavity, and after that morula is differentiated into the blastocyst. This blastocyst are beginning the implantation wall of the endometrium or endometrium layer of the wall of the uterus. This is morula. And these are the first event, the fertilization. This is the fertilization. Okay. Before I start fertilization, I would like to focus this figure. The uterine tube is start within the uterus to the lateral wall of the pelvic cavity. The lumen of the uterus is continuous to the uterine cavity. It must be memorized always. The uterine cavity, lumen of the uterine cavity is continuous with the uterine cavity, then cervical canal, and this is the cervix. So both sided, two-sided uterine tube, and they both are the continuous with the uterine cavity. Now the parts, gross anatomy of the fallopian tube. We mentioned the uterine tube start within the uterine cavity. So it is the first part is the within the uterus, that is interstitial part, and also known as the intramural part. And after that it is the isthmus, most narrow part, the isthmus, and from the isthmus to the infundibulum, it is the more longer and dilated part of uterine tube is the ampulla. And these are the region where meeting the sperm and ovum and culminate the mechanism or events fertilization. From the ampulla to the fimea, it is the infundibulum. And it is the luminal structure, so it has wall. This wall have three layers, importance: mucosal, muscular, and serous layer. Muscular layer, the smooth muscle have the ability to contract, so rhythmical contraction of the uterine wall importance both for the transport of the ovum and also the transport of the sperm. So these are mucosal layer. They have the gland, the secretion. They gives the nutrition. The result of fertilization, the zygote form, gives the nutrition, the zygote. And due to the continuous lumen with the uterine cavity, so there is formative zygotes gradually goes to the uterine cavity. Also the rhythmical contraction, and also there present cilia on the mucous layer. Their beat is unidirectional towards the uterus. Now, sperm cell movement. Sperm, where the sperm is naturally deposited in the vagina, and from the vagina the sperm start their journey to the fallopian tube. So first they overcome the cervical canal, then overcome the uterine cavity and enter the fallopian tube. And the region, ampullar region, the sperm is wait for ovum. Also, ovum comes from the ovary and enter the fallopian tube and wait in the fallopian tube, ampullary region for sperm. So, flow chart, the movement of the sperm from vagina, then cervical canal, then uterine cavity, then proximal part of the fallopian tube, and rest in the dilated part, ampulla. This figure showing the structure of secondary oocyte and sperm. This is mature oocyte or secondary oocyte. Structure of this oocyte, actually the covering of the oocyte and also the structure present in the cytoplasm. The oocyte, this is the pink color, this is the nucleus of the oocyte, and the present close to the plasma membrane cortical granules. These granules contain the lysosomal enzyme. These enzymes facilitate and help the sperm enter the cytoplasm of oocyte. The plasma membrane of the oocyte is covered by, surrounded by zona pellucida. This is zona pellucida, but the two structures are not fused. So there is a gap and known as the previtaline space. This previtaline space contain the polar body. So polar body, if questions where the polar body are present, they are present in between the zona pellucida and plasma membrane of oocyte. This space is known as the previtaline space. After that, most outer is covering is the corona radiata. Corona radiata cell of granulosa cell. This cells is also known as the nourish. These cells give the nutrition to the oocyte. These are the structure of the mature or secondary oocyte. This oocyte is present, the condition of the oocyte, the secondary oocyte, but in metaphase 2, that the metamucil 2 is incomplete. Up to the time the mucus 2 is incomplete because this secondary oocyte in metaphase, they present in the metaphase 2. Now the sperm. The sperm has definite shape and some exceptional characteristics. The sperm, this is the head of the sperm. The head contains the nucleus, and nucleus contains the chromosomal material, and this the cap, the acrosomal cap contains the acrosomal enzyme, acrosin, trypsin. These enzymes act during the acrosomal reaction. And this is the neck, contain the centriole. After that, the middle piece, there contain mitochondria, and this is the cytoplasmic process known as the tail or flagella. This gives the motility of sperm. Now, if the why oocyte is more valuable, or what is oocyte? Actually, oocyte is a cell, but it may be defined the divine mother of life, energy and knowledge. According to the law of biogenesis, they state that organisms arise only by the reproduction of other organisms, and the cell is responsible oocyte. And this cell actually gives beginnings of the life of a new human organism after an event known as the fertilization. So now we discuss the fertilization. By definition, fertilization is a process by which male and female gametes means sperm and ovum, fuse, then to form a large single cell known zygotes, occurs in the ampullary region of the uterine tube. This is the fallopian tube, and this is the ampullary region where the fertilization is start. Why ampullary region is chosen? Because this is the widest part of the tube and close to the ovary. How many days sperm remain viable in the female reproductive tract? For several days. How many? The number of the sperm deposited in the vagina, out of 1% only enter the cervix, and only one sperm are capable for fertilization of oocyte. Another question: all sperms are able to fertilize the oocyte immediately upon the arrival? No, are not able to fertilize. To acquire the capability for fertilized oocyte, sperm must undergo two conditions. One is the capacitation, another is the acrosomal reaction. So, what are the change, or what are the changes occur, or sperm must undergo two conditions. One is the capacitation, another is the acrosomal reaction. Now, what is capacitation? Capacitation is a period of conditioning in the female reproductive tract. Now, capacitation. Capacitation is the period of conditioning in the female reproductive tract. During this time, two structures, one is the glycoprotein coat and seminal plasma protein, that overlap the acrosomal region of the spermatozoa, should removed. This is the condition, and this is the capacitation of the sperm. This capacitated sperm can pass through the cell, corona cell, and undergo the acrosomal reactions. So changes is the removal of the glycoprotein coat and seminal plasma protein, which overlap the acrosomal region of the spermatozoa. This is the capacitation. After capacitation, the acrosomal reaction is occurred. In this reaction, there is a binding of sperm to the zona pellucida, and this reaction is induced by the zonal protein, and after that the release of the enzyme. This enzyme is needed to penetrate the zona pellucida, and the enzyme of the zona pellucida, the reaction, the enzyme acrosin and trypsin like substance comes from the acrosomal cap of the sperm. So acrosomal reaction in the acrosomal reaction, the main events is the some enzyme is released, and this enzyme is necessary. The acrosomal reaction, after binding sperm to the zona pellucida, this reaction is started and induced by the zonal protein. This reaction culminates in the release of enzyme like acrosin and trypsin like substance comes from acrosome of sperm. Now, phase of fertilization. What are the phase? There is a 3 phase. Phase 1, penetration of the corona radiata. Second phase, penetration of the zona pellucida, and third is fusion, plasma membrane of oocyte and sperm. These are the figure. This figure should try to draw and leveling is correct, and this figure is entitled not only the SAQ, also in the OSPI. So this figure every student should try to clear correctly drawing these figures. Drawing and leveling should correct. So how we can start this figure? First you draw the oocyte. This is the oocyte, this is the plasma membrane, and there is the oocyte present in the secondary second mucis, metaphase 2. This is the metaphase 2 stage. So this should focus, this should leveling and focus, and this is the previtaline space at the first polar body is present. After that, this is the previtaline space, and after that out they are surrounded by the zona. It is thick glycoprotein shell. So these are the zona pellucida. So this phase, the corona radiata pass, this is a phase 2, and these are after zona pellucida they are present the corona radiata. Corona radiata, the spiky arrangement of the granulosa cell. These are the sperm, they can try to penetrate the corona radiata. This is the first phase, and the painted the just second phase, and this is the third phase. So these are the figure, draw and level. Should try draw and level. Draw and level. The first the oocyte, previtaline space, zona pellucida, and this is the phase 1, phase 2, and phase 3. Now discuss on phase 1. Phase 1, penetration, corona radiata. 200 to 300 million spermatozoa deposited in the female genital organ. Out of them, only 300 to 500, not million, 500 reach the site of fertilization, and out of them only one of this oocyte sperm fertilize the egg. Previously we know discussed that the oocyte should capacitate. There is a condition, capacitation. After that, this sperm is able to penetrate the corona radiata. The second phase, penetration of the zona pellucida. This reaction, they can cross the zona pellucida, penetrate. There occur release of acrosomal enzyme, that is acrosin, and these are the reaction with the protein presence there, they can facilitate the reaction, and sperm penetrate the zona pellucida and contact with the plasma membrane of oocyte. When the contact in between the two plasma membrane, then the cortical granules release the lysosomal enzyme. They are present along the plasma surface of the oocyte. What happened after penetration the zona pellucida? The permeability of the, as soon as the zona pellucida changes their permeability. So they are impermeable other sperm. These are the very important events or step, because if the zona pellucida does not changes the permeability, the more sperm can penetrate the zona pellucida and try to enter the enter the cytoplasm of the oocyte. so their polyspermy may be done. So this condition prevent the polyspermy. And there is a question, what is zona pellucida? Zona pellucida actually the protein, glycoprotein shell surrounding the egg or oocyte, function that facilitate and maintain sperm binding and induce the acrosomal reaction. And both binding and acrosomal reaction mediated by the linked zona pellucida 3, that is a protein, zonal protein. Now phase 3, fusion of the oocyte and sperm cell membrane. Initial addition of sperm to the oocyte, this is mediated in part by the interaction of the integrins on the oocyte and the ligand and disintegrant on the sperm. So these are the then fusion both the surface of the oocyte and sperm. After addition, the plasma membrane of the sperm and egg fuse. Why they are fused? Because plasma membrane covering the acrosomal head cap disappear during the acrosomal reactions. Which regions is actually fused? Actually fusion is accomplished between the oocyte membrane and membrane that cover the posterior region of the sperm head. Posterior region of the sperm. The actual region, actual fusion is accomplished between oocyte membrane and the membrane that cover the posterior region of the sperm head. In human, both head and the tail of the spermatozoa enter the cytoplasm of the oocyte. This is the questions, Viva questions. In human, which part of the sperm enter the cytoplasm of the oocyte? Head and tail. So plasma membrane is left behind the oocyte surface. During these events, the oocyte responds 3 ways. That means as soon as the spermatozoa has entered the oocyte, the egg responds in 3 ways. First is the cortical and zonal reaction we discussed before. Now during this time, oocyte resumption of second mitotic division, and now they are metabolically active. It is surprising that activating factor is probably carried by the spermatozoa. The egg is very useful, very important cell, but their activation, metabolic activation, is help by the spermatozoa, because the spermatozoa give the activating factors. So both cells, sperm and oocyte, is important, and not only one cell can done this fertilization, so both cells are required for the fertilizations. These figures is total. This figure, there first is the phase corona radiata penetrate, second zona pellucida, and then fission. And this nucleus, this is the head of the sperm. It carries the nucleus. The nucleus is haploid, and the oocyte, this is the haploid. They are swollen and form the pronucleus. This pronucleus, male and female pronucleus fused and to form then after ultimately they form the single cell zygotes. There is a diploid number of chromosomes. This figure of the Langman, first these are the secondary oocyte, second there is a sperm enter, third there is a fusion of the pronucleus, these are single cell zygotes, and ultimately they they goes to the mitotic cell division, that is the start, and there's two cell stage form. So after 3 phase of the fertilization is complete, what are the result? The result of fertilizations, three result. First is the restore, second is the determines, third is the beginning. What is the restore? Restore the diploid number of chromosome. And now determine the chromosomal sex of the embryo, and last is the beginning. This is the pronucleus, contain the 23 chromosomes, and this is the pronucleus of the female, contain the 23 chromosomes. When they fuse to form 21, so chromosome restore, that is diploid. After that, this chromosome, if the male chromosome, they contain either X or Y, sex cell either X or Y. Now, response of oocyte during fertilization. During fertilizations, when the spermatozoa enter the oocyte, then oocyte responds in 3 ways. First, cortical and zonal reaction. We discussed already, this response is discussed. Now the resumption of second mitotic divisions that complete their second mitotic division, and last is the oocyte is metabolically active. It is surprising the metabolical activity of the oocytes. For that activations, the activating factor is carried by the spermatozoa. So this activity of those oocyte depend on the spermatozoa. This figure focus all the events of spermatozoa. This figure showing the total events of fertilizations. The first event, the sperm penetrate the corner radiata, second zona pellucida, third fusion. The total figures, these are the secondary oocyte and fertilizations. The sperm enter the cytoplasm, pronucleus fused and zygote form, and these zygote undergo the cell division and 2 cell stages already formed. After fertilization, the result of fertilization is very important. What are the result? This is the restoration, determination and beginning. Restoration of the diploid number. Pronucleus of the male and female. Male contain the either X plus 22 or Y plus 22, and female both are the X plus 22 and X plus 22. When the two pronucleus are fused, so 23 form 46 chromosomes. So diploid number of chromosome is restored, and determination of the chromosomal sex. In case of the female, the nucleus contain X plus 22 X and another is X plus 20. But sperm contain either Y plus 22 and either X plus 22. So the male pronucleus contain the Y plus 22 and female contain the X plus 22. They fuse. Ultimately, the embryo is the XY, that is male, and like this, if the XS, so XS the female. Now, beginning of the cleavage, that is cell divisions. First is the 2 cell stage, and there is a repeated mitotic cell divisions, the 2, 4 and 8 and ongoing. So these are the result. Result is restoration, determination, restoration, determination and beginning. Restoration of diploid number of chromosome determines the chromosomal sex of the embryo, determine the chromosomal sex of the embryo, and last is the beginning of the cleavage, that is cell divisions. Now the clinical correlate of fertilizations. We know fertilization, the process that from the zygote. This is a single cell from single cell actually beginning the life of humans. If they stop the fertilizations, so no zygote, no embryo, no pregnancy. And this method, either the fertilization will stop temporarily or permanently. This method is the, if we now clinical correlates. By the knowledge of fertilization, we will help the couple who maintain their family planning either temporarily or permanently. In temporary case, there are some barrier method. In case of the male, there is a male condom. In case of the female, the barrier placed in the vagina, including the diaphragm, cervical cap and contraceptive response. And these are the oral contraceptive pill. It is actually the combination of estrogen and progesterone hormone. These inhibit ovulation but permit the menstruation. So no ovulation, no fertilization, no pregnancy. This is all are the temporary. But there is also present permanent method. Tubectomy in case of female and vasectomy in case of the male. What is tubectomy? There is a ligation of the, first is the cut, the uterine tube and both end is ligate. This is the tubectomy. This is the tubectomy. And on the left side, this is the vas deferens. This is the pathway of the sperm from the testis to the exterior. If it cut this tube and ligate, this is the vasectomy. So the permanent family planning in case of male. Now, those couple are infertile. What are the measure? What are the treatment give this infertile couple? Infertility is a miserable life, and this problem occurs 15 to 30% of the couple suffering the infertility. This infertility result in case of the male insufficient number of the sperm and poor motility of the sperm. Because we know the normally ejaculate has 30 to 40 ml of the semen and approximately 100 million per sperm per ml. And males with the 20 million sperm per ml or 50 million sperm per total ejaculate, they also calculated the fertile. If there is a below, the person is infertile. Infertility in male, in female, in a woman, there is a lot of cause like this occluded oviduct, that is obstruction of the fallopian tube. This is caused, the most common cause is the pelvic inflammatory disease. They can obstruct the fallopian tube, and if their cervical mucosa is hostile, they prevent the transport of ovum. And spermatozoa is the foreign body, so there is immunity to the spermatozoa. And last is the absence of ovulation. What are the treatment delivered this infertile couple? First, they fertilize the outside the body, fertilize the outside, and after that it's placed in the ampulla region. So like this in vitro fertilization, gamete interfallopian transfer, zygote interfallopian transfer. In case the severe male infertility, that is few live sperm, the oligospermia, no live sperm, azoospermia, in this case, which treatment is preferable? Using intracytoplasmic sperm injection. Now complete the fertilizations. Next event is the cleavage. After fertilizations, zygote undergo rapid series of cell division known as the cleavage. So define the cleavage. Cleavage after fertilization. First replication, the after fertilization, the zygote form. This zygote undergo a rapid series of cell division. Rapid series of cell division known as the cleavage. So once the zygote has reached the 2-cell stage, it undergoes the series of mitotic cell division, so increasing the number of cells. And this cell is smaller in size and known as the blastomeres. Okay. This figure, first is a zygote, then 2-cell stage, then 4-cell stage, then multiple equation about the 3 days. After 3 days, the blastomeres compact the blastomere like is a ball of compact structure known as the morula, and after that they form a cavity. This is known as the blastocyst. So fate of the blastomere. Until the cell stage, they form loosely arranged clump cell. After the third cleavage, blastomere maximize their size and contact with each other, compact ball of cell held together by tight junction. Should remember, third cleavage, blastomere maximum size, tight junction, they held together by tight junction. And now there is a process compaction. It segregates the inner cell from the outer cell. And now they communicate extensively by the gap junction. Approximately after 3 days of fertilization, cell of the compacted embryo again divided to form 16-cell morula known as this is a marble-like structure, and this morula, inner cell of the morula constitute the inner cell mass and surrounding cells outer cell mass. Those are the inner cell mass give rise to tissue of the embryo proper, and outer cell mass form the trophoblast, which later contributes to the placenta. How blastocyst is formed? About the time the morula enter the uterine cavity, fluid within the cavity, they penetrate through the zona pellucida into the intercellular space of the inner cell mass. Fluid begins to penetrate when the morula enter the uterine cavity. So condition, morula enter the uterine cavity, and fluid can penetrate through the zona pellucida, enter the intercellular mass. Gradually intercellular space become confluence, and finally a single cavity is formed. This cavity is the blastocyst. At this time, the embryo is known as the blastocyst. So when the blastocyst occur, known as the blastocyst, but previously it is known as the morula. When the fluid enter and the space is become confluent and single cavity is formed, known as the blastocyst, and cavity is the blastocele. Now there is the inner cell mass and outer cell. The cell of the inner cell mass, now it is called embryoblast. At one pole, and these outer cell mass, trophoblast, flatten, and this is the epithelial cell covered the wall of the blastocyst. In this moment, zona pellucida disappear. and zona pellucida is disappear, it allow the implantation of blastocyst. So morula and blastocyst, main difference, morula has zona pellucida, was in the blastocyst disappear of the zona pellucida. First requisition for implantation, zona pellucida should disappear. Now implantation. There is this figure, this is the blastocele, two pole. Those pole, these are the one pole, they are present inner cell of the embryoblast, it is the embryonic pole. And this region, the trophoblast, over the cover of the embryoblast, this is the trophoblast. This region is touch the endometrium and gradually penetrate the endometrium of the uterus. So this is the embryonic pole and opposite is the abembryonic pole. So trophoblast cell over the embryoblast pole first begin to penetrate the epithelium of uterus. This is the first beginning. Which poles? There is a question. Which pole of the blastocyst can touch and embedding the endometrium? Embryonic pole. The which layer, outer layer, the trophoblast, they can erode and continuously embedding the endometrium of uterus. This is the embryonic pole. This is the trophoblast. They can touch and gradually embedding. So these are the embryonic pole, they touch and gradually embedding. So attachment and invasion of the trophoblast, some chemical structure involved. First is the integrin. This integrin expressed by the trophoblast, outer cell trophoblast, and there is another is the extracellular matrix laminin. Both are responsible for attachment. But there is another structure, a chemical structure, fibronectin, this responsible for migration, that is gradually eroding the endometrium. If we summarize the implantations, actually this is the result of mutual trophoblastic and endometrial actions. By the end of the first week, we say that is the development, the human zygote has passed three critical steps. First is the morula, then blastocyst, and last is the beginning of the implantation in the uterine mucosa. So what our three critical region is the morula formation, blastocyst formation, and beginning of the implantations. The zygote can pass three critical stages. So by the definition, what is implantation? Implantation is the process by which a blastocyst is embedding in the functional layer of endometrium, either anterior or posterior wall of the body of the uterus, usually close to the fundus, where it become embedding between the opening of the glands. This is the complete definition of implantation. The process, blastocyst embedding, where the functional layer of the endometrium, actual side either anterior posterior wall but close to the fundus, but away from the cervix, and in between the opening of the glands. During these times, condition of the uterus should know. Normally, we know the uterus has three walls from inside to outside. First is the endometrium, then myometrium, then perimetrium. Endometrium, the mucosal lining inside within the cavity, and then the thick myometrium, perimetrium, the covering of the peritoneum. Why this three layer condition of the uterus must know during these times? Because we can diagnose it. The lady is in the reproductive age, that is from the puberty to menopause, and endometrium is undergoes the regular cycle, maintain the regular cycle 28 days, and control of the hormone on the endometriums. But at the time implantation, mucosa of the uterus is in the secretory phase. This is another important question. During the implantation, the mucosa of the uterus is in what phase of the menstrual phase is present during the implantation of the uterus. It is the secretory phase, and during this time, the uterine gland, this is the uterine gland, and the artery become coiled, and the total mucosal layer is the edematous, succulent. This is the endometrial tube, this is the gland, this is the tortuous and also coiled, and this is the artery coil. And the mucosa, total mucosa is thick and it is edematous for reception of the blastocyst. What are the clinical correlation of the implantations? Normal implantations, endometrial functional layer of the endometrium, close to the fundus, any wall. But if the implantations is not close to the fundus, cervix abnormal, and isthmus intermuralis part of the uterine tube abnormal, isthmus abnormal, and also the ampulla abnormal, and this ovary also the abnormal, the peritoneal cavity. So abnormal implantation is known as the ectopic pregnancy, and at the different site where ectopic pregnancy may be done. So these are the different sites, either peritoneum, will be abdominal cavity, and also the ampulla of the fallopian tube, isthmus, intramural space. This any part of the fallopian tube, it is ectopic pregnancy, and either if it is the ovary also close to the cervix, it is also abnormal implantation. This is the clinical correlation of the implantation. These are the end of first week of the development. Thank you all. After complete this session, I hope my dear student should able for solving this problem, like some definition, ovulation, fertilization, cleavage and implantation, and question, formation, fate and function of corpus luteum, then what are the result of fertilization and also phase and result of fertilization, discuss in short of cleavage. In the two questions should practice the figure of fertilization, phase of fertilization, and also the figure of cleavage. Now the blastocyst formation and fate of the blastocyst, there is also include the figure. Write in short the condition of uterus during implantation. If you there is also mentioned, there is also drawing the figure. Now difference in between the morula and blastocyst, figure must, and what are the response showing the oocyte during fertilization. What is ectopic pregnancy and the different site. Some explanatory question, why sperm does not able to fertilize the oocyte during their first arrival in the female reproductive organ, and why the ampulla of the tube is site of fertilization. Thank you all. Allah Hafiz.